But now it is time for the man of the hour, Les Szabo. Dr. Les J. Szabo is a USDA ARS research geneticist and adjunct professor at the University of Minnesota Department of Plant Pathology. Les grew up around Seattle, Washington, and earned his degrees at Washington State University, and Michigan State University, and Oregon State University. He was a postdoctoral fellow at Rockefeller University before joining the USDA ARS Cereal Disease Lab in 1998. He was tasked with studying what was considered an obscure pathogen at the time, Vaccinia graminis, forma species Tritici. His early work laid the groundwork for genomics by showing that rust fungi have large, complex genomes and PGT has 18 chromosomes. This was followed by the first genetic map of PGT in the year 2000. In 2011, Les reported the genome sequence of PGT using Sanger sequencing. This was the first genome sequence of an obligate biotrophic plant pathogenic fungus known to science. Throughout his career, Les applied the latest technologies to studying PGT, starting with AFLPs and culminating with high throughput and long read sequencing via Oxford Nanopore and PacBio. Over the past 10 years, Les has been the go-to diagnostician for tracking the movement of PGT around the globe. Les developed a 3,000 SNP array for studying the molecular diversity of PGT and used this resource to develop diagnostic assays. Les is planning to retire December of this year. And Les, the floor is now yours.
Thank you, Matt. Let me share my screen here. So thank you, Matt, for the introduction, and thank you also for hosting this symposium and putting it together with University of Minnesota. It's been a wonderful set of talks. I want to thank each of the speakers for bringing a different perspective and showing the complexity of rust fungi. That's one of the things that has always fascinated me about rust fungi is its complexity. And you never know what you're going to find when you turn over the proverbial rock. So I wanted to kind of highlight a few accomplishments through the years. Many of these have been touched on by speakers already that in this session. But started off, as been mentioned, was that when I started 33 years ago and was tasked to bring molecular biology, molecular genetics to a rust fungus, we knew nothing, absolutely really nothing about its genome. And so we started out with answering very simple questions. What's the size and complexity of the genome? How many chromosomes? So I had a wonderful graduate student, Jan-Erik Backlund, who tackled the job of what is the size and complexity. And at that time, the tool that was used was reassociation kinetics. So you looked at how fast denatured DNA fragments reannealed. Now, a fun fact for anybody who has worked with rust fungi, Jan-Erik increased 48 grams of your dinispores, and he isolated 3 milligrams. That's milligrams, not micrograms or nanograms. That's milligrams to do this experiment. And he showed that it was a complex genome. At that time, there were 20 or so fungal genomes that had been looked at. Most of them were Ascomycetes. Most of them were quite simple. He showed that rust fungi were different. They had a large, his estimate was 67 million base pairs, and it was complex. It had lots of repetitive sequences. And so this gave us a start. I'm very happy to know that as this field went forward, we got lucky and we picked one that had a relatively small genome compared to many, many other rust genomes. But this was the first piece. The second piece was also done by a graduate student, Eric Bohm, in the lab, in collaboration with Dave McLaughlin and Bill Bushnell. And he asked and looked at the question as how many chromosomes. Now at that time, the literature based on looking at mitotic chromosome nuclei said there was 3 to 6. And that just didn't seem to match what the genetic data had been showing for rust fungi. And so we wanted to know what the number of chromosomes. So he did a karyotype by isolating single nuclei that were in packaging stage and serial sectioning them, and then doing 3D reconstructions from each of the serial section. And on the bottom, you see one of the maps he made. And he looked at 6 different isolates, and in each case, came up with a karyotype of n equals 18. The other thing that he showed was that the nuclear organizing unit, the NOR, where the ribosomal genes, was not terminal, but it was within one of the chromosomes. And so that's later important when one's doing genome assemblies, to know that the ribosomal repeat unit is going to be contained within a chromosome and not on an end. So also about that time, I was lucky to have Paul Zambino in the lab as a postdoc. And we got interested in phylogenetics. ITS sequencing and ribosomal sequencing was just starting to be used in basidiomycete fungi. And so we wanted to know about the relationships, phylogenetic relationships of cereal rust fungi. And so Paul did this initial study. It laid out our first picture of what the relationships are between the different rusts of wheat, Triticina, Striiformis, and Graminis, but also looking at within those groups. Now we had a suspicion, and others in the literature clearly suggested that what at that time Triticina wheat leaf rust was called Puccinia recandida was clearly a species complex. Cummings said that. And we showed here for the first time molecular data to say that indeed Puccinia recandida is a species complex and needed to be divided out into separate species, which it did later. The other thing I wanted to point out here and commend Amy on our great discussion on life cycles and changes in rust fungi is we also decided to look at transgenic law. And with Puccinia coronata, there's the correlated species Puccinia mesenterium. And it was predicted that those were going to be related. And so here on the bottom right-hand corner, we show this branch here. It has Puccinia coronata, and there are several different types. So as it is also was thought, it's a species complex, and Puccinia mesenterium belongs in this group. So working with rust, surprises come along all the time. And in this case, I got a sample from Steve Koke. He sent me samples of garlic rust that had devastated garlic production in California. So in 1998 to 2000, they had repeated epidemics of garlic rust. And they hadn't seen garlic rust in that Salinas Valley since the 1940s. And that was the heart of garlic production in the US. And the other odd thing was there were leek fields in the same area that weren't infected. And if you look in the book, Puccinia alii is a pathogen that infects garlic, but it also infects leek. So why are we having these infections on garlic and not on leek? And what's going on? And so we've said, sure, why not? So we started to work on PLPA also. And we collaborated with Yeshua Annixter and his team, Jacob and Tamar at University of Tel Aviv. The first thing we did was we sequenced the ITS region. And we showed that if you looked at the California samples, the garlic and the chives formed a discrete group. And the leek, and we had wild leek here from Israel, but we also, if you read the literature, in England, there was examples of garlic and leek rust. And so we clearly showed that there are two different types here. And so we wondered what was really going on. We had two distinct types. And so it did seem like it was just a mutation to overcome a few things. So Yeshua decided that he would look at the morphological characteristics. And he noticed, the other thing he noticed was that in the garlic, you saw only teliospores. So when he compared the morphology of the teliospores, the garlic type were smaller than the leek type. But then when he germinated it, he saw it made a proboscidium that only made 2 basidiospores, where the normal P. alii is, as Kathy talked about, it produces your 4 basidiospores in a typical meiosis. So we showed that this garlic type in California is actually a hemicyclic type. It only makes teliospores and basidiospores. And so the difference here is a life cycle variation. So we also meandered some more, and there was a soybean rust outbreak. And it had creeped up from South America and was in 2004 was first found in the US. And there was interest in trying to get some molecular analysis on it. And I had Charlie Barnes as a postdoc in the lab. And he first developed an assay, a nested qPCR assay, where he could detect down to a single equivalent of a single Uridinea spore in a rain sample. And so we thought that a great way to look at movement of P. packerizii spores is to look at rain deposition or wet deposition. And so initially he made some traps, but then through Professor Kruger at the Plant Path, we got connected up with this National Atmosphere Deposition Program, which was set up in the 1970s to look at acid rain and air pollution. And they have hundreds of sites across the US. And they are interested in the chemistry of the water. And they throw away the particulate parts. So we hooked up with them and we looked at between 70 to 121 sites across 4 years to look at spore deposition. And then we worked with Scott Iser to model infection and basically the amount, the risk of infection in soybeans by the amount of spores being transported. And so in the bottom here, you see an example of a model for the 10th through the 16th of September and the amount of spores that would be predicted. The dark red counties are the sites of infections. And then you see the dots are spore traps that we collected and were able to pick up. In this case, these color represents moderate amounts of spores. And so here we're combining rain deposition, qPCR, and modeling to try to provide a predictive model for risk for soybean rust. Now, back to cereal rusts. One day, Marty Carson came into the lab. He had been looking at barberry bushes up by the student center on campus, and bushes that were supposed to be resistant to P. graminis had ECL infections. And so first thought was, oh no, resistance is broken down. So he gave the samples to Kim in my lab, and we had a qPCR assay to look for Puccinia striiformis, Puccinia graminis, Puccinia, and so she ran that assay, and she came and told him, it's not Puccinia graminis, it's Puccinia striiformis. And I was at a meeting at the time, and he said, no, that can't be right. Go do it again. But then diligent work by Eugen and sequencing work by Kim and others in the lab showed that indeed this was Puccinia striiformis, and that we had solved the century-old mystery of what the ECL host, or gametic host, as Kathy calls it now, is for Puccinia striiformis. So then back to genomes. So whole genome sequencing was beginning. We were fortunate to get a grant from NSF to sequence Puccinia graminis. And working with Christina Kumo at Broad Institute, we were successful. We were generated a genome assembly. We collaborated with Sebastian to do comparative genomics between the two, and came out with this first paper based on Sanger sequencing. And as John clearly talked about earlier, shortly thereafter, working with John Fellers and Xiaomen Qing and Goose Baccarat, there was a comparative genomics between the three wheat rust pathogens. And John pointed out some really nice work on the synteny. And I just wanted to highlight also one of the things that came out in that paper was the first characterization of the mating type locus. I mean, we really know very little about the mating type system in rust fungi. So then UG99 happened. And we've heard good explanations by Dave and Hovmoller, Moens, and others about UG99. And when I first heard 1990, Alan Rolfs and Don McVeigh talking about this new isolate from Uganda that had virulence to SR31, I wouldn't have predicted that this would become a major part of my program for the rest of my career. And Dave did a good job of talking about the surveillance programs that's been developed over the years and the diagnostics we've done. And the point here is, as Dave made and Moens has made, it's all done because of collaborators. And I just show a few pictures here of the wonderful collaborators from around the globe that I've been able to interact with over the last decade or so as we worked on UG99. And then, as Dave also mentioned, and Moens, that pretty soon our focus shifted. We weren't only interested in UG99. There were new guys in town, and that doesn't mean Eugen and Pablo. There were new rusts that we needed to worry about, and we need to understand their relationships. And so we started developing the tools to understand phylogenetic relationships between the different races, and could show that in many cases, you have the same race in a different genetic group. And so you really need the combination of genotyping and phenotyping to really understand these shifty enemies. So here I am nearing the end of my career, and I'm working on genomes again. And as mentioned by two of the speakers, we're in a new era of genomics using long-read technology and being able to phase haploid genomes. And as Melania explained real nicely about their discovery of UG90 evolution through somatic hybridization, and how that came about by sequencing 2 isolates and looking at the data and realizing, wait a minute, these guys share a haploid genome. So we were interested next to say, okay, can we identify the other haploid genome donor that's involved in the evolution of UG99? So we went back to our phylogenetics, and in all the phylogenetics, clade 2 is always a sister group to clade 1. So that looked like it should be a good starting point. Let's look at clade 2 to see if indeed that's the second nuclear donor. And so we chose this isolate, this 87Ken3018-1. It's an isolate derived from a collection in 1987 in Kenya. And initially what we did was we sequenced a number of conserved genes. And we used Oxford technology so we could sequence the whole gene in a single run. And so we could separate them out into haplotypes for that gene. And what we soon found out is that we could use a set of 8 genes to be able to look at, predict, and make predictions about haplotypes. And those 8 genes said indeed clade 2, our 87 Kenya isolate, had the other haplotype found in UG99. So it had the C haplotype. The other thing that we had discovered by going through the resources at the Serial Disease Lab is we went through old isolates from Africa looking for progenitors to UG99. And we came up with we found an isolate that was collected in 1972 in Ethiopia that is a member of the UG99 lineage. With our genotyping, it's almost identical. So we had 2 pieces of this puzzle that was laid out by Melania and Peter. We had a good candidate for the other donor, nuclear donor, and we had an ancestral isolate for the UG99. So we then did PacBio sequencing, and we used hi-fi sequencing, and working with Adam Herman at the University of Minnesota in the bioinformatics, and the postdoc in the lab, Radhika Bartatula. We assembled, Adam was able to develop a pipeline where he could get a almost completely phased assembly of these two isolates. The number of contigs in the initial assembly, the paired assembly, were in the low 20s in the number of contigs. And so he was then able to assemble these into the 18 original chromosomes. And we have a map here of showing those 18 chromosomes, showing our isolate and the two haplotypes, A and C from 72 Ethiopia 11-1, and the two haplotypes from 87 Kenya, C and D. The dots on this figure represents telomeres. We found that the ends of the contigs would have large telomere repeats in the range of 300 to 500 repeats. And so we have highly contiguous, yet in some cases, end-to-end assemblies for these two haplotypes. Also did comparisons where we looked at conserved genes. So we did dot plots, which Melania showed. And so on the top, A and B, we're comparing haplotype A from our 72 Ethiopia 11-4 isolate with the assembly of 21 not haplotype A that Melania talked about. And you can see by the nice diagonal line, there's collinearity between the assemblies, and it matches very well. We did the same thing with the haplotype C from the 72 Ethiopia and the 87 Kenya, and you can see that indeed those are identical haplotypes. If you then do the other comparisons, where you take haplotype A from 72 Ethiopia and the 21 not, you can see that they are collinear, but there's a lot of divergence. And you see the same thing if you compare haplotype C and haplotype D. Another way to look at this is to do a heat map. So Adam made this heat map, which looked at pairwise divergence of these conserved genes. And you can see here divergence, the dark red is there's no divergence at all. And so as expected, if you look within a haplotype, whether it's from UG99 isolate or 72 Ethiopia or the 21 not, the haplotype A, it's all, there's essentially no divergence in these 2,000 conserved genes. And then you see the same thing for in haplotype C and so forth. And then, but the other thing you do is you begin to get an idea of relationships. You can see that haplotype C up here is more divergent to haplotype D than haplotype A. Okay. So my career has spanned 33 years. I started it working on genomes, and I'm ending it working on genomes. But as they say in a lot of things, it always takes a community. And research is no different. It takes a community. And I have been very, very fortunate to be part of a very active and engaging community at the Serial Disease Lab for my 33 years there and the University of Minnesota. It's been a pleasure to work with this outstanding group of people at all levels of science, and students and postdocs and administrative people. I wanted to highlight in the 33 years, these are the scientists at the CDL that I've worked with. It's kind of amazing that how stable the number of scientists have been. I want to highlight really specifically Bill Bushnell here on the left and Alan Rolfs on the right. They were my mentors when I arrived at the Serial Disease Lab. I was a molecular biologist. I had worked with bacteria, and closest thing I worked with was Canada albicans, Ascomycete fungus. So they were my mentors, and I owe them a great deal. Of my success, I owe to these two outstanding individuals. And then one is also not successful unless they have good people in the lab. And I've really been blessed over my 33 years to have a stable lab environment. So I've had 3 main technicians over those years. So on the top left is Jamie Mork, and then Alexi Balmuth and Jerry Johnson. And they have been the core and the life of the lab for so many years. And then also 2 people I want to really highlight is Kim, who's been there for a long time, and she's been a key person to the lab. And more recently, Nick has joined the lab and is doing wonderful things and helping me finish out my career, hopefully on a high note. And then I need to thank the Rust community, especially the serial Rust community, who have been so welcoming and thought-provoking and supportive over these many years, and people I've developed real friendships with. They will always be a soft place in my heart for all of them, and I look forward to staying in touch with many of them in the future. And on that, thanks.
Thank you very much, Les, for the inspiring presentation and summary of the last few decades. And it's a really heartfelt talk, and really great to see. You know, I know a lot of you are spread out around the world, so you're welcome to open up your own beverage of choice to celebrate Les's retirement as you like. And I don't know, Les, if you're able to stay on a little bit longer to maybe address some anything in the chat, or panelists who would like to chat further. But I believe that more or less concludes this symposium. So any last words from you, Les?
Just once again, thank you, Matt, and thank you to the wonderful group of speakers, and showing the wonderful complexity and the uniqueness and the fun that one can get from studying Rust fungi.
All right, well done, Les. And thank you all for your participation and attendance.